Hi, Michelle Glass here. We are finally ready for the tricky part of our Chapter 8, Part 2. Our topic for this video is meiosis. So we're going to take a little bit of time here to really go through this in detail. Make sure that you're taking good notes and you have a nice artwork in your textbook if you want to refer to that instead of trying to draw your own. Meiosis, we've talked about, is our division for the production of gametes. Gametes are your egg and sperm cells. We can describe meiosis as a reduction division because we are going to be reducing our number of chromosomes from diploid to haploid. In the case of humans, that means from 46 chromosomes to 23. So you're halving it, but you're dividing your number of cells. So you're going from 1 cell to 4 cells. We've also talked about meiosis as sexual reproduction. How can we take a single diploid cell and divide it into 4 haploid cells? Well, we are going to take that cell through 2 different divisions. We're going to call those divisions meiosis 1 and meiosis 2. Now, each one of those divisions is very similar to what we did in mitosis. So meiosis 1 is going to have prophase, metaphase, anaphase, telophase, but we're going to indicate them as prophase 1, metaphase 1, anaphase 1, and telophase 1. So PMAT 1. And then we're going to do it again for meiosis 2. So meiosis 2 will have, again, the cell going through, this time we have 2 cells going through prophase 2, metaphase 2, anaphase 2, and telophase 2. So it's PMAT 2. Now, essentially, the steps are the same as what happened in mitosis, but there are a few important key differences that you need to make sure you pay attention to as we go along in our conversation. Here's one of the key differences between mitosis and meiosis. Meiosis is producing 4 unique haploid daughter cells. And if we look back at our notes from mitosis, remember you're just getting 2 identical cells at the end of mitosis. Okay, so when we talked about mitosis, we did our cell cycle and we said that the cell is going to prepare for division during that phase called interphase. And that same thing is going to be true here. So the cell still goes through interphase as part of its cell cycle. In interphase, that DNA is in its relaxed chromatin form, and this is where your DNA replication is going to occur. So remember, you're going to start out with homologous chromosomes, and it's going to go through DNA replication, and you're going to end up with your sister chromatids, which are identical to each other. From there, we're going to see that the cell goes through prophase 1. Now, prophase 1 is very similar to what we had in mitosis. Your nuclear envelope disappears, your spindle apparatus forms. This is actually when the DNA condenses to form chromosomes. But here is something special. In prophase 1, we have that crossover event happening. And we've talked about that in a separate video. In the crossover, we get the formation of recombinant DNA. So you get a combination of information that you've inherited from your parents showing up that you can pass on to your offspring. This can happen, this is random, it happens differently every meiosis, every prophase 1. It can happen more than one time in your homologous pairs. But remember, like in the case of humans, it's not happening with those sex chromosomes. Then we see the chromosomes are going to line up in the middle of the cell for metaphase 1. But here is something else that's new. They stay paired. We did not see that in mitosis, but we see that here. And then we are seeing here also that independent orientation where each pair lines up independently of each other. So I've just drawn one pair there in the cell, but if I draw a second or third or fourth or fifth, I can organize them in different ways, lining up down the cell. And then we have our anaphase 1, and so we see here that the homologs, the homologous pairs, are going to separate in anaphase 1. Then we have our telophase 1 and cytokinesis, and there's not a lot of new stuff or special stuff that's happening there. You're just going to get the formation of your 2 new nuclei. Cytokinesis is your 2 new daughter cells. Now those cells don't stop. They're going to do it again. We're going to have another round of prophase, metaphase, anaphase, telophase. We see prophase 2 is basically the same. So we're going to get the formation of our spindle apparatus. Our DNA is condensed into chromosomes and our nuclear envelope disappears. Metaphase 2, those chromosomes line up in the middle, but we don't see any kind of pairing up happening here. Anaphase 2, sister chromatids separate. And then telophase 2 and cytokinesis, we end up with our 4 haploid unique daughter cells, and they're unique because of the crossover event that's given us 4 possible combinations instead of just 2. Also, we could, if we're representing this with more than 1 pair of chromosomes, we could get different outcome depending on how we line them up because of that independent orientation. So there's lots of room for diversity here during meiosis. Crossover is one of our important sources of diversity. So we've talked about this many times. That formation of that recombinant DNA has given us 4 possible chromatids instead of just 2 possible chromatids. Independent orientation, again, is adding to our genetic diversity because we're going to get in humans, we have 8.4 million combinations of how our chromosomes can end up in the gametes. And then it's even more diverse than that because of crossover event. And then the last bit of genetic information, genetic diversity, is coming from random fertilization. So again, there's so much diversity that's happening in the female in the production of her gamete. There's so much diversity that's happening in the male in the production of his gamete that then we don't know which one of those sperm is going to fertilize which egg that was formed. And so that's given us a third layer, a third source of genetic diversity. So this is why sexual reproduction is so important for organisms, because of all of these different sources of diversity, allowing our populations to have differences so that when environments change, we still have individuals who are able to adapt and survive. And that's it for now.